Good day. I'm here actually to continue from where we stopped last time on this important chapter, which is called sterilization and disinfection. Remember last time when we met in class, I introduced you to the topic where actually I described different terminologies that are associated with this context, which include disinfectant, bactericidal, bacteriostatics, germicidal, and so on and so forth. So related to that, also I classify actually the sterilizations. Actually, what I mean, the sterilization, all these uh, different categories that actually emanate from this important context. So from there, we understand that we have different methods of sterilizations, which include thermal methods and non-thermal methods. So due to the actually nature of the thermal, so it took a lot of time for us to be able to thrash out all that you need to know on that particular area, which we managed to discuss the what you call the sterilization by heat uh, and sterilization by heat in form of moist heat and sterilization by heat in form of dry heats. So what we didn't discuss is the non-thermal methods of sterilizations, and that is where is going to form the basis for discussion today. Okay, so among these non-thermal methods, based on the classification I have given you last time, we have those classifications by those methods actually by the physical methods and those methods by the chemical. Even by physical, we have two types, okay, which include radiation methods and then filtration methods. Then the chemical methods include using chemical agents, which mostly we use the gaseous chemicals, okay, which we'll discuss later. So actually that is just a brief background just to recap what we have discussed last time. So without further explanation, I'm going straight to the topic that we need to discuss today, which is sterilization by radiation, which falls under non-thermal physical methods. Okay, so by the word itself, radiation, okay, you should be able to understand that we are using some form of radiations. In this case, we use ionizing radiations, which include X-ray, gamma rays, electron beams, okay, which are very popular actually in a lot of biomedical applications. One of the most popular applications that you can remember is chemotherapy, uh, radiotherapy. Radiotherapy is a form of treatment that we use by actually trying to uh, irradiate the cancerous cells using ionizing radiations. So based on the concept of ionization of this kind of energies emanating from radiations, the radiation can be able to actually disrupt the DNA of the cells and thereby leading to the death of the cells. So we can apply the same concept, or the same concept are applied in this case for sterilization. But instead of actually the tumor, in the case of radiotherapy, so in this case, we are actually targeting the bacterial cells. So radiation can be used actually effectively to destroy microorganisms such as bacteria, viruses, fungi, and including their spores, including the bacterial spores that are highly resistant, okay, which actually have a kind of a preventive uh, measures in addition to the normal vegetative bacteria. It has a kind of reinforced cells walls, cells walls, which actually prevent or minimize its vulnerability to certain agents or certain variables such as high temperature and certain chemicals. Okay, so these radiations that I mentioned, which include X-ray, gamma, etc., can actually be effectively utilized to destroy not only the not only the pathogenic microorganisms, but also including the bacterial spores. So radiation have little effect on biological organisms because of genetic and biological changes that actually occur. As I mentioned, the damaging effect of radiations will depolarize, mutate, break, and alter structure of DNA, which actually ultimately lead to the cell's death. The deaths may occur due to the formation of toxic molecules like peroxides, uh, one of the other reasons in addition to the ionizing effect. So this actually indicate that we employ radiations not only for radiotherapy as in case of cancer treatment, but rather we can also use it for sterilization actually to mix our pharmaceutical product free from any form of pathogenic bacteria and their spores. Not only bacteria and many other microorganisms such as fungus, such as viruses, etc. Then the next category under this physical sterilization method is what we call non-ionizing radiations. Okay, so in this method of sterilization, actually long wavelengths, low energy, and low penetrating radiations are used for destroying the microorganism. In contrast to ionizing radiation, which we use actually wavelength with radiation which has a longer wavelength and which has high energy and high penetration power. So in this case, we use the ones actually that are not ionizing and which have actually low wavelength. So the method is usually employed for sterilization of substances, okay, and it actually includes infrared radiation and ultraviolet radiation. You remember UV spectrophotometer, right? Okay, UV spectrophotometer emits this kind of actually uh, rays actually that have this capacity. But in that case, we are using it for the UV spectrophotometer. It is actually being used for analytical purposes. But we have another UV equipment which we use actually for sterilization. That UV sterilization equipment emits certain rays of UV light, which actually has the capacity to interfere with the normal functions of the microorganisms system and thereby lead to their death. So the wavelength of infrared rays is longer than visible light and it is low energy electromagnetic ray. So one of the examples is the infrared rays that kill microorganisms by the process of oxidation of important microbial microbial biomolecules that are used for sterilization of a lot of equipments and apparatus. Among them, we Among them include syringes, catheter, and so on. So the wavelength of UV radiation is shorter than that of the visible light. That is something that you have to remember. They are always shorter than that of the visible light. And the wavelength of UV light emitted approximately a value of 253 nanometer, which is considered as a germicidal rays. Okay, so this UV radiation is not generally considered to be a good sterilizant. Rather, it is employed in the reduction of microbial load. So most of the industries that utilize this method actually do not actually rely solely on the UV to achieve sterilization, but rather it is occasionally being employed as a secondary method to achieve effective sterilization. Okay, when you compare it with other methods, so this may consider as less effective, but it can always be actually utilized as a secondary or as a supplementary method that can be used to achieve complete sterilization of a product. For example, in the water treatments, recently I was in the factory with students, we are seniors, for example. One of the plants that we visited, they use in their water treatment system, um, they use reverse osmosis system. But in some of the tanks, okay, during the process, above the tanks, that they have this equipment that releases UV rays that actually supplement or actually supplements the effects of the filtration sterilization that they use in order to achieve complete sterilization of the water system that they are using to produce pharmaceutical product at that facility. So this is a way of just corroborating the system of sterilization instead of relying only on the filtration, but also UV is added as a supplementary after the filtration. So that is it. In living organism, many cellular materials, which include nucleic acid, actually they absorb this UV light, and when this happens, it results in the formation of what is called pyrimidine dimer, which is a bonding of two adjacent pyrimidines, and that usually lead to the inhibition of replication of DNA. So if DNA is halted, when the replication of DNA is halted, that may actually temper or interfere with the reproduction of the bacteria and normal bacteria or any other microorganism, and that actually may lead to disturbance in the normal function of the microorganism system. And this will eventually lead to the death of the microorganism, or they are unable to reproduce. Okay, so UV light are effective for the contamination of surfaces, water, and air. As I have cited the example, I recently observed one of the pharmaceutical plants using this UV lights as a supplementary sterilization method for their water system. So that it is considered as one of the effective method that can be utilized to actually get rid of microorganisms from the water, among other places. UV lamps are usually installed in rooms like aseptic rooms, microorganism laboratory, large equipments, air ducts, operation theaters, laminar flows, among others. These are just some of the examples of the UV light sterilization method. So that is for the radiation method for sterilization. Then the second physical method of sterilization is what we call uh, filtration. Filtration, I'm sure, is not a new concept to you. Yeah, the UV may be relatively bit unpopular to you, although you know it. Maybe you by you know UV application uh, uh, in the pharmacy by other actually missions or by other objectives such as analytical analysis, but we also use it for sterilization. So, but in this case, filtration actually is something that I consider all methods of actually trying to separate things, right? So when we say separation, like you separate some substances, usually in liquid, we are actually using sterilization as a separation techniques, right? To separate certain substances that are contained or present in a liquid. So remember, this kind of methods actually is only effective for certain fluids, whether it's liquids or gaseous. Okay, so it is a method of sterilization using that is usually prepared in case of thermolabile solutions. Thermolabile solution, those solutions that actually can get uh, can be destroyed when exposed to high temperatures. So that means the thermal methods is not an option for sterilization those substances. So in that case, we have to employ non-thermal. One of the most effective is what we call this filtration. So you can see how it works is that usually the substance in solution that are sufficiently small to permeate across a membrane, a membrane, what we call a filter membrane with perforation, okay, can permeate through. While those substances that are large, their size is larger than the perforation of the filter membrane, cannot be able to permeate through. So by so doing, we can be able to retain microorganisms on the surface of the filter, because the filters are designed in such a way that they have the capacity to retain, okay, all the bacterial cells, okay, and only allow the maybe some molecules that are desirable, okay, some ionics that are desirable to permeate through the other side of the filter membrane. So this process actually is in such a way that microorganisms can be physically removed by absorption on the filter media, as shown in this illustration. Okay, you see the bigger black dots, they are the ones representing microorganism cells. Okay, so they are actually physically retained on the surface of the filter media. Okay, so by so doing, actually the solutions, the other content of the solutions, maybe some drug content in ionic form or in molecular form, can only be the one to be allowed to pass through the filter media. Okay, so by sieving from solution and gaseous without exposure to heat. Okay, so this devoid actually using thermal method that can actually have certain deleterious effects on the solution. So this is sometimes also called a cold sterilization because it does not involve any heat. It must always be kept in mind that the filters shall not be interfere with the solution or gases, neither by adding unwanted component nor by removing essential constituents. Okay, so the development of filter media has to be carefully done. We cannot use substances that are active, that have the ability to absorb or leach out their component to contaminate the solution we are processing. So that's what this statement is saying. We have various filters. We have various filtration systems that we use to achieve this kind of mission of sterilization by filtration. Okay, so one of the um, uh, filtration system that we use include membrane filters, millifo filters, synthetic glass filters, cis filters, backfilled, and Mandela filters. Okay, so these are the ones that I highlighted here, which I will discuss briefly in the coming slides to understand how to appreciate how is our work in order to help process our samples. So most of them actually work based on these mechanisms. You can see this illustration is trying to show that on the left-hand side here, we have a solution that is contaminated with bacteria. Okay, you can see the cells of bacteria right in here. In addition, these are the cells. They have some kind of branches, some kind of branches that help them to swim within a certain medium. But on the other hand, we also have certain cells. These are maybe certain proteins, certain ions, certain solids that are desirable, that are that are needed to be present in the solution. So when we use a filter media, this is a filter media here. So all those microorganisms here, they cannot be able to permeate because the size of the population is well controlled in such a way that it cannot allow the cells to pass through. And on the right hand side, after the filtration, we have a sterile solution where only the desirable uh, substances or components of the solution are allowed to be in it, and the solution is devoid from content of any microorganism. So that is how it works for all of these filters. Although they have different features, which we are going to learn in the coming slides. So starting from the first one, membrane filters. By the word membrane, it has a membrane, right? So membrane is just a straightforward word that I don't need to elaborate further. So what these filters actually are made of cellulose acetate and nitrates, okay, and polyvinyl chlorides, nylon, polycarbonate, Teflon, and polysulfonates. These are some of the substances or materials that are used to prepare the membranes that actually do the job. So the full diameter of these membrane filters ranges from 0.2 micrometer to 0.02 micrometer. Okay, so usually they come within this size range and they are suitable for sterilization as the size range of the bacteria and viruses varies from 0.3 micrometer to 0.5 micrometer and 20 nanometer to 0.36 micrometer respectively for the bacteria and the viruses. Okay, so accordingly, based on that, certain bacteria that are identify, especially the bacteria, because when you can see from this range here, is mostly suitable for bacteria because virus they are much smaller. They are much smaller. So because we have viruses that can actually be in the size of up to 0.02, which is actually uh, uh, below the size of the perforations of the filter media. So they are mostly suitable for bacteria because their pore size ranges from 0.02. Okay, so for the viruses, we have other filters that can do that job. The characteristics of solvents in pharmaceutical preparations is important as they affect the filter. Such a problem does not arise where water is used as a medium. Yeah, because we have different ingredients or raw material that we use that can be present in different types of solvents. So we have to make sure we choose the right filter medium for a particular solvent. In other cases, due to development of membrane filters with highly resistant material, solvents do not create any problem. Yeah, for some other types of filters. To obtain a sterile products, membrane filters, including all other accessories, shall be sterile by suitable method before the commencement of filtration, and the entire process requires aseptic condition. So these are physical substances. The membrane filter I've shown before, which I will show in the next slide. Okay, they are components. So those components actually had to be in sterile form. Has to be in sterile form before the commencement of the procedure. Because if they are contaminated, they have some form of bacteria. Those contaminated agent can be in the solution we are trying to treat. So first of all, the filter medium, the container that is going to be utilized to retain the process solution, the panel and all other associated gadgets has to be in sterile form before commencement of the procedure. And the process, the entire process has to be done in a sterile environment under aseptic condition. Since the filtrations through this flow is very slow, vacuum is applied to increase the rate of filtration. The filter, the membrane filter is used for sterilization of ophthalmic solutions. Ophthalmic solution, those kind of medicine that we use for the treatment of eye condition, eye drops rather, some eye drops, ear drops, and so on. Antibiotics, biological preparations, hormones, and a wide range of solutions that are heat sensitive. Particles larger than the pore size are physically trapped, as I explained before, and those that are sufficiently small can actually permeate through. Therefore, other than porosity, uh, in addition to that physical way of retaining the bacteria, actually electrostatic attraction also takes place. Okay, so electrostatic attraction of particles also can occur on the filter. So other than the porosity, the efficiency of the filtration is also dependent on other factors which include electric charge of the filter. So the filter media actually, some of them has certain charges which also play a very vital role in accumulating or retaining the microorganism cells and prevent them from passing through. So electric charge carried by the organisms and the types of fluid being filtered are all considered as variables that actually play a role in the efficiency of the filtration. So this is the membrane filtration system. From the left hand side, you can see the membrane filter here. This is how it looks. We have some of them in the lab right there. I think if you want to see, you can ask the lab technician to show you one. So this is how it looks. We have some that are in loose form that we use in certain settings. Like in this case, you can see them here. So this is the zooming on how the membrane filters look like using those material. Remember the cellulose, Teflon, and so on that we use to actually manufacture the membranes perforations. And this is the complete unit. This is the complete unit. So this you can see the unit involve various tools which are used actually to eventually achieve this sterilization. This is the container, the bezel, the kind of plugs which actually contain the process solution, that is the sterile solution. We have a rubber stopper here, we have a stainless base, we have the main one that do the magic, the filter membrane at the middle. Then on top of it, we have a locking ring that actually prevents permeation or spillage of the material that is being processed. And we have a after that locking ring, we have a funnel that we actually use to uh, accommodate the solution that is being filtered. Okay, so you can see something like this. This is just a schematic diagram. Okay, we have an equipment, electric pump, which actually do the suction. It suck uh, it provides a suction so that the filtration can be done within shorter period, because the perforations is so small. So if you just allow it to 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 take place under normal condition, it takes time that cannot be actually, that is not realistic to do the experiment. So we have to provide this suction in order to do the process in a reasonably accepted time. Some of the advantages of membrane filters include ability to be utilized to process thermolabile solutions. Absorption of medicating is negligible as the membrane is very thin, so it's one of the advantages. They don't absorb, they don't waste your solution. The rate of filtration is reasonably rapid, and large volume can be filtered quickly. A new disc is used for every new preparation, thus the chance of contamination is very less. So the bezel can be changed at any time that is so desired, but the drawbacks will include uh, process is limited to liquid and gases only. Okay, unlike other methods that we can utilize to process equipments, okay, apparatus such as process, such as whatever, container, clothing, and so on. So this one we can only process gases and liquid fluids, whether it's in liquid form or in gaseous form. One of the limitation also is that the filters are expensive, as new disc is required every time. Another disadvantage include fine force may get clogged easily. Okay, sometimes we experience that problem, they clog. So when that happens, actually they have to be replaced. Then chemically, these filters are less resistant and are soluble in certain organic solvents, for example, ketones and esters, which means you can use them to process certain corrosive reagents. Next category of filter is what is called millifo filter. Okay, you can see by design they are similar. Okay, they are similar with a membrane filter, but these ones they are called millifo because they have different features. Let's learn the features they have. Millifoil filters have specific pore size and are made of pure and biologically inert materials like nitrocellulose and polyether sulfone. Okay, so you can see the composition or the material that we use actually to prepare them is different from that of the membrane filter. These are available in full size ranging from 0.2 micron to 12 micron. Okay, so which means they are a little bit bigger in size, say, compared to the membrane filters. The full size of millifoil filter is relatively uniform, exhibit high thermal stability, does not absorb the solutions, and it is resistant to chemicals. You see, one of the key also advantage of this one over membrane filters is they can be utilized to process even uh, corrosive chemicals, certain chemicals that you cannot be able to process using the membrane filter, you can use this one to process them. Then the next one is what is called sintrate glass filters. Sintrate glass filters are made of borosilicate glass and finally folded into a desired size and fused together by heating to a sintering point. Okay, so these filters are made actually with a borosilicate glass, which is which is grounded in a powdered form and then fused together using a heating process. So temperature at which the adhesion of the particles take place is important. Okay, so certain temperature are used actually to fuse the powdered glass together and form a membrane, which is called discs. So the discs of the appropriate size and shape is then fused into Paris glass funnel, okay, having the size of Bochner funnel. And this fused glass filter have different pore size. Filtration is generally carried out under reduced pressure for the removal of bacteria solution. So this is how these central glass filters look like. This is how they look like. This is the borosilicate glass that is powdered and then fused together at high temperature to form what is called a disc. You can see it looks like a tablet. So, and this tablet actually now is attached. It's actually uh, get affixed in a special glass that looks like a Buchner glass, and that forms the basis for the filtration. So it's something that can be reused. It's all like the previous filters, membrane filters, and millifoil filters that are actually not supposed to be reused. In this case, actually, it is designed in such a way that it can be reused again and again. Okay, so this is how this how it looks. This is how the system looks for this central glass filtration unit. We have the main one, which is called central glass filter. You can see it here on the top. Okay, then we have the receiver, the suction plugs, which we use actually to receive the filter. So the materials or the solution to filter is poured through the central glass on top, and then the filter media, which is central glass filter, will actually do the job by retaining the microorganism cells above and then allowing the spew solution to permeate through. And usually suction is provided using a pump. Okay, you can see this is where the pump is connected here, and then when the machine is switched on, it provides a suction which leads to high speed filtration rate. Okay, so that is how this one works. So remember, before commencement of any of these filtration operations, the equipment, all this equipment has to be in sterile form. Okay, that is the only way you can ensure that whatever you process is filtered, is actually uh, sterile. What are the advantages of these central glass filters? It can be, it is actually, it can be filtered by these injectables, which we call parental preparations. They can be utilized to, they can be processed by this filter, uh, as all the foreign particles as well as microorganisms can be filtered out. Okay, so in addition to, when we are talking about uh, parenteral solution, especially those that are injected, right? When we say parenterals, we are referring to those solutions that are meant to be injected, and that means direct into the body system. So in addition to the requirement for absence of microorganisms, there's also requirement of absence of any foreign particles. So these central glass filters, they are a good choice in trying to achieve that goal. Okay, so it's one of their advantage. It is also suitable for small and large volume solutions. The amount of filtrate retained in the medium is negligible. They don't retain much, so they don't waste your solution. The disadvantage include its impart alkalinity to the filtrate, which may cause precipitation of the alkaloids from salt solution. So that is one of the drawbacks, because they are manufactured from borosilicate glass, so they have that possibility of giving imparting certain alkalinity. So if the solution is desired not to be alkaline in any way, so this is not a good choice. Another disadvantage includes loose fibers of the asbestos may shed from the parts. Okay, so there's a risk of shedding of some of the fibers uh, uh, to get into the processed solution, and it is not suitable for filtration of small quantity of solution, as asbestos uh, asbestos parts absorb a considerable amount of solution of the API. Okay, so it is also one of the disadvantage of these particular sintered glass filters. The next filters are what we call SEES filters. SEES filters is a bacteria filter which is based on filtration of asbestos pass filter. Okay, this is asbestos pass filter similar to that for the sintered glass filters. So in this one, we call it a filter disc with a force so fine that will not permit passage of bacteria and allow solutions to emerge uh, in the receiving end as a sterile solution. Okay, so you can see they are very similar with the previous one. They are very similar with the sintered glass filter. The only difference is that while the sintered glass filter, the filter disc is permanently fixed in the funnel. In this case, you can see there's a clip here, there's a clip here, okay, which are called clamps. Then the asbestos actually are added in between. So meaning the asbestos can be replaced as when needed, okay, without discarding the complete funnel. Unlike in the other case for the central glass filter, it is fixed. So that is one of the difference between the two. Otherwise, the construction is the same. Okay, the construction is the same. So let's elaborate more on construction. It consists of two parts. The lower part pitted with a perforated plates over which compressed asbestos part is placed. Yeah, this is the lower part, okay, meaning the base of the funnel, okay, and the upper part has a value through which the pressure can be applied, okay. Both parts join together by a wedge nut, so this one and this one, and then the valve is present on the upper part through which vacuum is applied, okay. So this is how the system looks like, okay. This is how the system looks like, very similar to central glass filter. um, it also has some advantages and disadvantages. One of the advantages of this filter is that it doesn't have risks of contaminating the filtrate, unlike the central glass filter that can alkalize, that can make the uh, medium more alkali. So this one is actually neutral. It doesn't have that disadvantage. The apparatus is very simple to use. For viscous solution, they are more suitable. So meaning it can be utilized to process the viscous solution. This is how the filtration units looks like, okay. One of the difference, very clear difference between this one and central glass, you see the previous one is actually made of glass, but this one is made of stainless steel. You can see it is made of stainless steel, so it is actually can withstand a lot of pressure because it is highly resistant. So the filtration also can be done rapidly. It can be done rapidly. So the disadvantage is that asbestos may shed loose fiber. The part may absorb some amount of medicament. The asbestos part may yield alkali and causes precipitation of alkaloids. So that means even this one has that chances of alkalizing the medium. Okay, so both central glass and cis filters, they have this disadvantages of making the medium more alkali. So they share more common disadvantages. Okay, when you compare between the two. It's just that this one actually is lesser in terms of that disadvantage when you compare to the central glass filter. The next filter that I want to discuss is what is called Backfield filter, right? Backfield filter, also known as candle filters. They are also filters that are utilized to sterilize various solutions. Okay, so they are actually also called ceramic filter because they are made with porcelain or kiselgu with different porosity. The medium is made of ceramic, so that's why they are also called ceramic filters. The the filter process the filter process in the candle filter is used by the candle, which actually is a tube shape. The tube or cylindrical shape filter is accumulating the flow from outside to inside. Okay, that is how it happens. I'm going to show the image in the next slide. So it allows the actually flow of fluids from the outside, the contaminated solution from outside will have in contact with the outside spaces, while the filtered sterile solution will be actually received in the interior part of the filtration unit. The narrow opening of the one end of the candle is attached to the vacuum, and the candle is placed in the solution to be sterilized, and filtration is carried out under reduced pressure. So the entire process is carried out under aseptic condition, and candle is sterilized by moist heat sterilization before commencement of the system. So this is how it looks. This is the candle. This is the candle. This is how they look. There are multiple of them in one unit, attached in one unit. So it is designed in such a way that this is the PET. You can see this is actually how the instrument looks like, the intact instrument. The PET, meaning the contaminated solution, will actually pass through this opening. So when it gets into this portion, so this is the candles, different candles that are mounted in the filtration unit. So the solutions will actually pass through the candles, which actually do the job of filtration here. You can see the exaggerated image of the candles here. Okay, so the filter solution will actually get through the candles and then come out from here. Okay, from here, and whatever is here on top is considered as a sterile solution, and this is where the filtrate is drained out into another container. So this is how it looks. You can see the machine, and this is the X-ray. This is the intact machine. This is the X-ray of the machine, and this is the elaboration image of the candles, and this is how it works. So over time, as the operation continues, there will be a cake on the outer surface of the machine. So the cake actually uh, is dislodged by actually pumping a kind of air, a compressed air through the filter, or some air or certain sterile fluids. Okay, that is filtered to that one is actually injected from the interior component of the tube, that will lead to dislodgement of the cake that actually clog the filter media. So this is how it works. It also has advantages and disadvantages. The advantages include uh, that it is suitable for sterilization of thermolabile solutions. Okay, those that cannot withstand heat, they can be utilized. The candles can be utilized to process those solutions. Uh, Two, living as well as dead microorganisms can be removed by this. So both living and dead, yeah, can be actually controlled using this kind of filter. The disadvantage include force of candles usually get blocked and require cleaning from time to time. So as I shown before, we have a procedure that we can utilize actually to unclog the filters, but it's one of the advantage because the pores are so small. Okay, so they can easily get clogged. Arrangement of the filtration units is difficult, and chances of contaminations are more when you compare to other filters, because this filter is also meant to be used again and again. It's not something that you can just replace. Hopefully, when you like millifold filters or membrane filters. So that is for the physical sterilization method. Then moving to the chemical sterilization method. In this case, we use what we call gaseous sterilization, which is considered as uh, one of the methods that had been utilized very long ago as well. In those days, in the olden days, we had a chemical agent called formaldehyde, okay, which is which has a very little effects on microorganisms, that has been utilized actually as a agent for sterilization using this method. But nowadays, actually, this chemical agent formaldehyde is not being used. It is restricted. due to its high reactive nature and difficulty in removal from materials. So on the basis of that, on the basis of the safety concern, it is no longer being utilized as a agent for chemical sterilization. So what we are mostly using nowadays is what is called ethylene oxide and beta propiolactone. Okay, they are the ones that are mostly being utilized nowadays to do sterilization by gaseous methods. So ethylene oxide is considered as the most widely used gaseous sterilization agent. It is the most widely used, okay, especially in the pharmaceutical industry. It is being used when the option of using gaseous sterilization is required. It is simple, ether at room temperature. It is a gas, highly inflammable, and become explosive when mixed with air. Yeah, so it has to be handled with care. It can easily, meaning it can easily catch fire. Okay, so this kind of sterilization agent actually has to be handled by experts that know what they are dealing with, because fire is one of the big disasters that must be prevented. Okay, so it is a gas that is highly inflammable. It can easily catch fire if not handled properly. So that's why proper dilutions has to be made. Okay, proper dilution with inert gas such as carbon dioxide or fluorinated hydrocarbon actually is necessary. And this dilution will lead to making it non-inflammable and safe to be used. Commercially available ethylene oxide contain 10%. It contains 10% ethylene oxide with 90% of carbon dioxide. Okay, so highly diluted, only 10% of it. And the lethal effect of ethylene oxide on microorganisms is due to alkylation of essential metabolites, uh, probably by replacing active hydrogen on sulfhydryl, hydroxyl, carboxyl, amino groups, and hydroxyethyl radicals. So by this chemical reaction, actually, when this occurs, that leads to the death of microorganisms. Sterilization by ethylene oxide requires a proper validated procedure where specific conditions, that is relative humidity, exposure time, and temperature, are to be maintained. So all these three variables, relative humidity, temperature, okay, and exposure time, has to be controlled. They have to be set at a desired range. You can see the ranges they are given for each. For the humidity, has to be in the range of 40 to 50%. For the exposure time, has to be considerably longer time, minimum 1 hour. Okay, and for the temperature, it has to be about 55 degrees Celsius for this sterilization to be achieved. And the concentration of ethylene oxide that is being used for gaseous sterilization ranges from 200 to 1000 milligram per liter. Okay, so that is the concentration for effective sterilization to be achieved. Any concentration lesser than 200 milligram per liter will not be able to be effective in eradicating or destroying microorganisms. The increase in concentration of ethylene oxide and temperature reduces the exposure time. Yeah, so when the intensity of the chemical and the temperature is high, it is considered at the time that is required to achieve complete sterilization may not necessarily have to be high, it has to be reduced. In general, concentration of ethylene oxide, exposure time, and temperature should be 450 milligram per liter, okay, 6 to 16 hours, and 49 to 60 degrees Celsius for temperature, for for the concentration, temperature, respectively. Okay, so this is how. Okay, I missed something here because this 450 is referring to the concentration. I also missed time. Sorry, I have to edit this slide. So what this values represent the concentration, and this value represent the exposure time, and this value represent the temperature. So I mean to write uh, for concentration, uh, exposure time, and temperature respectively for proper sterilization to take place. So these are the optimum parameters that you have to actually take note that must be achieved for the proper sterilization to be achieved. Okay, that means at the concentration of 450 milligram per liter of the ethylene oxide under the exposure of 6 hours to 16 hours and at a temperature of 49 to 60 degrees Celsius, okay, for a proper sterilization to take place. After sterilization, the separation of ethylene oxide from the material is essential and is done by aeration. So we don't want traces of this chemical agent in our processed products. So that's why the aeration, since it is airborne, so the aeration of the solution is done to make sure it get right of all the chemicals out of the sterilized solution. But remember, this aeration is done actually in under aseptic condition, and the air that is being utilized for the aeration is also sterile air. It's a pure sterile air. So what are advantages and disadvantages? For the advantages, the substance are capable of withstanding high temperature can be sterile using this method, meaning thermolabile substance, although uh, the substance capable of withstanding actually, so meaning that they can only be utilized to process non-thermolabiles. Okay, thermolabiles are vulnerable because temperatures of 55 is considered as a bit high on certain substances. Okay, surgical instruments such as catheters, plastics, disposable syringes, needles, etc. are effectively sterilized using this method, meaning they can be used to sterilize certain equipment, medical equipment that we need. Powders packaged in polyethylene bags can be sterilized using this method. Moisture-sensitive substances can be sterilized without deleterious effects. Okay, so these are some of the things that we can utilize, that we can utilize this method to process. Disadvantage include due to relative longer exposure time, the process is slow, meaning it is lengthy and it is not suitable for emergency. You see, from the ranch I've given minimum of 1 hour. So if actually the process needs to be done uh, in a kind of speed in a very short period, so this one is not a good method in emergency situation. Then it is highly inflammable and may cause danger if proper precautions are not taken. So that is one of also the disadvantages. Then the next category of chemical disinfectants, which is also gaseous, is what is called beta propiolactone, BPL, as an abbreviation. BPL is a non-inflammable, unlike the ethylene oxide. This one is non-inflammable, and chemically, it is a cyclic lactone. It exerts a strong bactericidal effect against different varieties of microorganisms. It is widely used for sterilization of rubber goods, plastic material, and delicate optical instrument, and also used for sterilization of plasma, inactivated viruses, vaccines, etc. Because of its vapor pressure, because of its vapor, poor permeability, it is used it is actually uses its uses are restricted. Okay, its uses are restricted to sterilization of only large surfaces like a big premises, homes, etc. So because it has a kind of a high BFO effects, okay, and the permeability is considered as less when you compare to ethylene oxide. It doesn't permeate certain small areas, so it uses it's restricted in only sterilizing premises that are bigger in sizes, such as certain environment like rooms, corridors, and big equipments. So this comes to the end of the sterilizations methods that you need to know as far as this course is concerned. So let's quickly move to the last one, related topic, which is called disinfection, okay, which I define at the beginning of the chapter. So disinfection, just by way of recap, it's actually a process of using a chemical called disinfectant to destroy, inactivate, or significantly reduce the concentration of pathogenic agents. When we see pathogenic agent, we are referring to microorganisms that we don't want, undesirable microorganisms that causes problem on our cells and on our products, such as bacteria, viruses, fungi, and so on. So from the surfaces of inanimate objects, okay, so that is one thing that you have to remember for disinfectant. When we say disinfection, we are referring to a process of actually using a disinfectant to destroy, inactivate, or reduces the burden of pathogenic microorganisms from the surfaces of inanimate objects. Okay, so disinfectants are utilized on inanimate objects. Okay, like a kind of tools which doesn't have life. Okay, we don't use them on animal, on human or animal surfaces, because they have a tendency to actually uh, uh, cause some damages on the tissue. Okay, they are corrosive in nature, and some of them are toxic, so they cannot be applied on the surface of the skin. So another associated term close to this one that can be utilized on the skin is what is called antiseptic. Antiseptics, right? Sepsis, so it's a process of actually cleaning uh, the the premise or area of animals or humans. So that is by way of definition. You can see from the image, these individuals, they are disinfecting this environment. So we get to know about this now. So you can see disinfection is a process of using disinfectants, right, to actually control uh, microbial contamination. So what are these disinfectants? Disinfectant is a chemical agent which reduces the number of vegetative microorganism, either by removing or destroying them. Okay, so disinfection is different from sterilization. Sterilization, in addition to destroying vegetative microorganisms, they also destroy the bacterial spores, non-vegetative ones, which are called spores, okay, that are dormant in nature. So for disinfectant, largely, there are some that have such a capability of destroying the spores, but a lot of them, they don't destroy the spores. So that's why that is one of the major difference. Although some disinfectants are sporicidal, meaning they have the capacity to remove the spores, however, a chemical does not need to be sporicidal to be classified as disinfectants. Okay, So that is one thing that you must to remember. A disinfectant that kills spores is sometimes described as sterilant or chemosterilant. Okay, so it's one of the terminologies that we use to describe this disinfectant that actually be used for sterilization. So what this definition is trying to show you is that all sterilant or all chemical agent for sterilization are disinfectant, but not all disinfectant are, are sterilizing agents. Okay, disinfectants of varying formulation have been used since the late 19th century. This is there is a range of different types of them. There is a range of different types of these disinfectants, and their formulation they are formulated in different ways. Okay, with different actually content of active ingredients. This brings us to the types of disinfectant that we use. We have different types of them. Okay, depending on the content of the active ingredients, they have different mode of action and different efficacies. So first of all, one of the category of disinfectant is what we call sanitization. Related term, sanitization is the disinfection and cleaning of an area or an item. The term sanitizer is normally taken to be an agent that both cleans and disinfects, normally a disinfectant that contains a cleaning agent. Okay, so this is one of the category of a disinfectant that are called sanitizers. Sanitizers, they are just chemical agents which are used actually to control, okay, to limit or to reduces the concentration of microorganism. But usually, uh, while you are limiting the concentration, you are also cleaning the surface. You are removing the bacteria as well as certain deaths, certain undesirable particles and deaths that are actually on the surfaces. So uh, in addition to the disinfectant that the disinfecting agents that they contain, sanitizers contain certain agents that are used for cleaning. For example, detergents. Okay, they contain certain detergent, certain surfactants that are that can actually repel or dislodge undesirable particles on the surfaces of substances. You can see from the image here, this particular individual is using sanitizers to actually clean the surface. While they are removing the microorganisms, they are also removing the unwanted particles that are called deaths, right, on the surface of this object. So that is one of the category that you must remember. We use sanitizers to clean in addition to removal of microorganisms. Some disinfectants are classified as microstatics, to what we call bacteriostatics. I defined that previously, so I don't need to elaborate on that, where the ability of bacterial population to grow is halted, right? Bacteriostatics that so says they only halt the reproduction. They only halt the reproduction, but they don't kill. Bacteriostatic disinfectant can cause selective and reversible changes to cells by interacting with nucleic acid, inhibiting enzyme, or permeating into the cell wall. Once the disinfectant is removed from the contact with bacteria cells, the surviving bacteria population could potentially grow. So that is one of their drawbacks. As the exposure of the chemical is halted, is stopped, the bacteria will continue to regenerate. The microorganism will continue to regenerate and continue to cause the undesirable effects. Other disinfectants are called in contrast to the bacteriostatics, right? This category is called microcidal, which is known as bactericidal in case of bacteria, in that they destroy bacterial cells through different mechanisms. How does they do that? In this one, they are not only stopping the reproduction, but they are destroying completely. So this includes causing structural damage to the cell, autolysis, cell lysis, or the leakage or coagulation of the cytoplasm. Okay, so that's how they actually works to destroy the microorganism. Within these groups, the spectrum of activity varies, with some disinfectant being effective against vegetative, gram-positive, and gram-negative microorganisms only, while other disinfectants are effective against even fungi. Other disinfectants have a broader spectrum and are called sporicidal, in that they cause the destruction of endospores forming bacteria. Okay, so we have these categories of bacteria. Remember, I mean disinfectants. We have under disinfectant, we have sanitizers that contain uh, uh, actually cleaning agents such as detergent and all that detergent and soaps, they are put in addition to the chemical agent for killing the bacteria. We also have microcidal and microstatics, which actually control statics. They just stop the production and replication of the microorganism, while the cidal they actually kill through various mechanisms. So under this category also of cidal, we have broad spectrum, okay, which actually can kill spores. And that name also, once they have the ability to kill even the spores, they are called sporicidal. Okay, sporicidal disinfectant, meaning they can be even utilized to achieve sterilization. There are a number of disinfectants available in the market with different modes of activity and of varying effectiveness against microorganisms. There are various approaches to the categorization and subdivision of disinfectant, which include grouping by chemical nature, mode of activity, or by microstatics and microcidal effects on microorganisms, as I said before. And the two principal categories are division into oxidizing and non-oxidizing chemicals. So the majority of non-oxidizing disinfectants have specific mode of action against microorganisms, but generally they have a lower spectrum of activity compared to the oxidizing agents. So non-oxidizing agent actually they are non-effective as oxidizing when compared to oxidizing agent, and when compared to the spectrum of the microorganism that they actually control. So some of the example of these non-oxidizing agents include alcohol, just normal alcohol, okay, quaternary ammonium compounds, and phenolics. Okay, so they don't oxidize the cells of microorganisms, but still they are being utilized actually for control of variety, number of microorganisms that are unwarranted or undesirable. On the other hand, the oxidizing disinfectants are chemicals that generally have non-specific mode of action against microorganisms. So they have a wide spectrum activity. They're non-oxidizing disinfectant with most type able to damage endospores, but they pose greater risks to humans' health. Okay, so actually, the non-oxidizing ones they are considered as more safer, and that's why they are even some of them are even utilized as antiseptic. You see, most of the a lot of antiseptic that we use, sanitizers, hand sanitizers, uh, and so on and so forth, we use this alcohol, right? Because they don't cause much destruction or damage on our cells. But these ones, the oxidizing ones, actually they are more lethal in terms of destruction ability to microorganisms, and they actually uh, prompt more danger to cells. So they are not recommended for being used on actually skin or animal part. So they have some of the example of these non-oxidizing disinfectants include halogens, parasitic acid, and the hydrogen peroxide, which we use actually to disinfect premises, environments, and all our areas of works. So with this, I think we have come to the end of this particular uh, uh, topic for sterilization and disinfectants. And this marked the end of the portion that I supposed to put you through as far as this course is concerned. So thank you for your attention. We'll see again soon. Bye bye.